Experimental models that closely resemble the human lungs are critical for enabling scientists to study lung health and disease. Inhaled air enters the lungs through the trachea and primary bronchi. Inside the left and right lung, these primary bronchi branch further into increasingly smaller airways. The smallest airways are called the bronchioles and end in the tiny air sacs called alveoli. The respiratory tract is lined with epithelial cells, which are the first cells that come into contact with what we inhale. The epithelial layer differs in its cellular makeup in the various regions of the lung, and epithelial cells can be separately isolated from those regions. After harvesting, the epithelial cells can be grown and maintained outside the body using a laboratory technique known as cell culture. Cell cultures are traditionally performed in 2 dimensions, where the cells are grown on plastic dishes completely submerged in cell culture medium. However, the lung is such a complex organ that standard cell culture methods alone cannot capture all its complexities. Over the past decade, there have been several advances in epithelial cell culture methods that have allowed scientists to better mimic lung tissue. This includes the introduction of air-liquid interface, lung-on-chip and organoid culture models. The research groups at the Leiden University Medical Centre and the Erasmus Medical Centre, Sophia Children's Hospital, are actively involved in the development and implementation of these advanced culture methods. In the air-liquid interface model, lung epithelial cells are cultured in inserts on porous membranes at an air-to-liquid interface. This model mimics the composition in the human respiratory epithelium and allows scientists to study exposure of the cells to airborne substances. The lung-on-a-chip model is a small micro-device that comes in various forms and shapes. Within the chip, lung epithelial cells and cells from blood vessels can be co-cultured across a porous, flexible membrane and are exposed to a flow of nutrients in one compartment and air in the other. By stretching the membrane on which the cells are cultured, it's possible to mimic the mechanical forces of breathing. The organoid culture model is essentially a 3D engineered simplified mini-tissue that is formed from stem or progenitor cells embedded into a matrix to allow the cells to self-assemble as they would do in the native organ. These models are powerful tools for studying lung health and disease more reliably compared to traditional models, including testing new drugs, testing the toxicity of inhaled substances, and will allow scientists to find new ways to regenerate damaged tissue.